I just took a DNA test, turns out.
That Lizzo wouldn't be able to do so without the amazing advances in DNA sequencing technology that we have today? Over the past few decades, the cost of DNA sequencing has exponentially decreased, while its efficiency has exponentially increased. But all of these advances wouldn't be possible without the Human Genome Project. Let's take a look at the process that made this possible, known as Sanger sequencing, or chain termination sequencing. Here's how it works. First, you'll take your DNA sample. Then you'll take some DNA polymerase, which is what creates a complementary strand of DNA during DNA replication. Of course, you'll need your building blocks, your dNTPs and your ddNTPs. A dNTP, or deoxynucleotide, is basically like one singular nucleotide, like an A, T, C, or G. And a ddNTP is almost the same thing, except the two has a key difference, which lies in that extra pesky D in its name, making it stand for dideoxynucleotide, which means that it has 2 instead of the usual 3 oxygens. So back to our DNA polymerase. As it goes along, it creates this complementary strand and all is well, until it tries to put on that ddNTP. At first, everything's fine. It connects to the previous nucleotide, no problem. But the issue comes when you try and put on a nucleotide after your ddNTP. The two can't actually bond because the ddNTP is mixing that extra oxygen needed, thus terminating the chain. Hence why the Sanger process is also called the chain termination process. The DNA polymerase will move on and keep creating little strands like this that stop at each ddNTP, and each of these are flagged with a fluorescent marker, making it easy to see under UV light. When you repeat this process across all 4 bases and run it through an electrophoresis gel, they'll be separated by mass. With the help of a UV light, you'll be able to see your glowing DNA sequence. Of course, Sanger sequencing isn't exactly the most efficient, but it still has its advantages even today because of its high accuracy and low costs. However, next-generation sequencing is able to handle a lot more data a lot faster. In sequencing by synthesis, you can have millions of clusters running at once, and in sequencing by nanopore, you can even have your DNA be sequenced in real time. It's with these innovations that we're able to do all the things we can with DNA today, such as analyze the genomes of COVID-19 and other diseases and track their evolution, or take our own DNA tests and figure out who we're related to at pretty low costs. And of course, it's given rise to a pretty iconic song by Lizzo.